Welcome back to our PCR product series. Now that we have covered qPCR and RT-PCR, let's learn about PCR reagents. By using strategic protein engineering, ABM scientists have developed many different DNA polymerases for various application needs. Each of our enzymes has its own attributes, for example, high fidelity, high selectivity, or high speed, and together they provide a comprehensive collection for our customers. All the enzymes and buffer formulations are developed and manufactured in Canada. We perform stringent quality control for each batch as part of our standardized procedure to ensure customer satisfaction. Customers can then choose the best suited DNA polymerase based on the properties and characteristics of the enzyme using this table. We offer the 2x master mix format for most of our DNA polymerases. This means that the customer will only need to add in template, primer, and water to finish the setup of the reaction. The use of master mixes provides better experimental reproducibility and simplifies the reaction setup. Our master mix with dye is pre-mixed with DNA loading dye. Therefore, after the PCR reaction, the PCR product can be directly loaded onto an agarose gel. A good DNA polymerase should have the ability to amplify different DNA templates under various conditions. When compared with the other competitors, our BestTac DNA polymerase is able to give a better product yield with shorter extension times. This impressive elongation rate of 3 kilobases per minute offers flexibility in the end user's experimental protocol. For example, if the amplicon size is 1 kilobase or less, only 1 second would be needed for the extension time. Even with the shortened extension time, the results are not compromised, and as you can see, it is able to amplify a range of DNA templates ranging from 100 base pairs to 100 kilobases with high fidelity. One of the reasons that our BestTac polymerase is able to amplify a range of DNA templates is that it can tolerate many commonly encountered PCR inhibitors. This attribute is highly valued for blood samples, where oftentimes a separate DNA purification step is required before the PCR reaction itself. By utilizing our BestTac polymerase in our blood direct PCR lines, we find that it not only eliminates the need for DNA purification in blood samples, it can actually tolerate as high as 12% of blood volume in the PCR reaction, outperforming other competitors on the market. Apart from blood samples being difficult due to the vast amount of PCR inhibitors present, other difficult DNA templates can result from having secondary structures or high GC content. When tackling difficult templates, a robust DNA polymerase formulation like our Kodak polymerase is required. With its unbeatable robustness compared to 2 other competitors, it can handle templates that other polymerases cannot amplify. Even though its speed is not as fast as BestTac, it has the same fidelity and overall offers a strategy for stubborn templates. Plant samples are known to be difficult templates for PCR, and they usually require optimization with multiple enzymes and protocols. We combined our Kodak 2x PCR master mix with plant gDNA extraction buffer to make plant research easier. Having a robust polymerase eliminates the need for PCR optimization. The process involved extracting the gDNA from plant samples in just 20 minutes, after which a standard PCR protocol can be used to amplify the gene of interest. To take our product offerings to another level, ABM has developed our PCR SureKit for optimizing difficult PCRs. With years of experience in PCR product research and development, we have an extensive knowledge base of PCR additive and buffer formulations. The PCR SureKit includes 12 number-coded PCR master mixes, each with a different buffer system containing different PCR additives. A customer can first try amplifying the difficult target with all 12 master mixes, select the best performing master mix number for that particular target amplicon, and order the particular number-coded master mix for further PCR studies. For routine cloning applications, we recommend our BestTac DNA polymerase. Kodak DNA polymerase can be used for problematic targets that BestTac cannot handle. Finally, try with PCR Sure if both Kodak and BestTac cannot amplify the target. Based on our in-house statistics, this BestTac Kodak PCR Sure rundown has an over 99% success rate. Downstream analysis of PCR requires agarose gel electrophoresis to separate the amplicons by size. The commonly used DNA visualization agent ethidium bromide, also known as EB for short, is biohazardous and constantly poses a health risk to researchers. Our solution to this health threat is to offer a series of EB alternative DNA visualization agents, the SafeViews. The SafeView series is a group of DNA binding dyes that combine to nucleic acids. SafeView Classic and SafeView Fire Red are gel casting dyes, whereas SafeView Plus is a post-electrophoresis staining agent. ABM also offers EB alternative gel loading dye, including Safe Green, Safe Red, and Safe White. These are 6 times concentrated gel loading dyes, meaning that the customer just needs to mix PCR samples in a 1:5 ratio and load the mixture onto a plain EB-free agarose gel. The operation details are the same as EB for DNA visualization, which makes it really easy for the customer to switch over and replace EB completely in their labs. Besides reagent consumables, ABM also carries all the plastic consumables needed in PCR experiments. We have all different formats of PCR tubes, plates, and films. Shopping for your PCR consumables was never this easy. ABM is your one-stop shopping center for all your research needs.